Our last speaker of this session is Ed Acevedo. Ed is Professor of Kinesiology and Health Sciences and Associate Dean for Graduate Studies in the College of Humanity and Sciences at Virginia Commonwealth University. His research focuses on physiological responses to mental stress and the potential crossover benefits of physical activity. He will be speaking this morning on the physiological activation to acute mental challenge, implications for cardiovascular health. Ed.
Thank you. Thank you. It's quite an honor to be speaking today, and I want to start by not only thanking the program committee, but my academic advisors, Diane Gill and Alan Goldfarb, who are here, although it looks like Alan just left. I'm wondering what that means. So let's jump in. One of the thoughts that I've always had is that maybe the brain and body are connected. And maybe if they are connected, we ought to be able to see that in a number of ways. And I would argue that maybe we should see it at a cellular level. Now, to get there might be pretty hard, and it's actually taken me about 25 years, but the kind of understanding that we have about how maybe the brain and body might be connected actually starts with our own appreciation of how we experience anxiety. It's a very simple model where if you get nervous or anxious, all of us can describe what that feels like. It might feel like your palms are a little sweaty, like mine. It might feel like your heart is beating a little quicker, like mine. And we might actually share all of those experiences as humans. Interestingly enough, the public media has gotten a hold of this, and we all appreciate it in kind of our culture. And we connect it with things like heart disease. But if we look at the literature, it's not quite as clear on how these things are connected. One of my favorite books, and a book that I require my students, doctoral students, to read before they come to the lab, should they finally choose to come to the lab, is actually by Robert Sapolsky. I don't know if anybody has read some of his work, but he's a biologist at Stanford. And he's done a great job of taking the literature, scientific literature, and using it to describe how it is that we function as humans and connect that to other species, and takes an evolutionary approach as well. One of his favorite areas is actually anxiety and stress. And this book is clearly my favorite, but all of his books are brilliant in how they make science real. He really is interested in the biology of behavior, and that actually is what our interests are as well. The themes that I'm going to be covering today, we're going to talk a little bit about history. We'll talk about actually the stress response, and we actually understand that pretty well. I'm going to link that to physical activity and stress and that relationship. And then we're going to go into what I've most been interested in, and that's the immune system, the inflammatory response and stress. Because for us and for me in our lab, we're interested in how it is that the brain might get all the way to the cell, and if it does, how that might impact heart disease. And last, we generally use acute stress. We'll combine some stresses so that we have an idea of how we might have a titration effect of stresses. And you'll see how we do that in the lab. We play a couple of games to see if we can look at, let's say, low stress, moderate stress, high stress. And this is extremely difficult with humans, although others in the past have done it a little bit better than we are allowed to do it today. And then we switch and we say obesity is actually a chronic inflammatory state. So we can take obesity as a chronic stress and then add an acute stress in the lab. And we try to do a lot of things in the lab. If we look acutely and then we propose or infer the longitudinal effects, Cannon was the first to write about the stress response in particular, although Darwin talked about it in terms of survival. Cannon was the first to coin the term the fight or flight response, right? So when we're stressed, we're ready to do something physical, to flee or to fight for survival. This has been critical in our evolution. Hans Selye came around and said, well, maybe it's not all good. And so he had a three-phase aspect to the stress response. And he suggested that this exhaustion phase was problematic and led to disease, right? And his was called the general adaptation syndrome. That general part actually was not quite accurate as well. He thought all the systems responded the same, and we know that that's not exactly correct either. Then we had Roddy, and he's one of my favorites because he began to do a lot of human research and try to really understand what it is that humans are experiencing physiologically when we stress them. So I'll tell you a little bit later about some of the protocols he used that unfortunately we can't use anymore in trying to understand this. Then I'm going to go towards the benefits of physical activity in this stress response. And that's been proposed initially by Richard Deanspear. John Cacioppo has done a huge amount of work on emotion in health. His whole career, a tremendous amount of investigative work there. Then a group of us have tried to connect physical activity again for the last at least 20 or so years. And then we'll get into new frontiers, but I'll move the slide quickly before you read those. If we look at the central nervous system and the stress response, it's activated. We all know that, but we're really talking about this aspect of the activation of the central nervous system. We're really just looking here. We also know that once we look there, there's the sympatho-adrenal medullary axis and the hypothalamic pituitary. I don't want to go down there, right? But we know that we're activated, and we can see that in cardiorespiratory responses, and we can measure hormones in the blood, stress hormones, epinephrine, norepinephrine, two favorites, and cortisol. And we know that we're going to have a response at the cellular level, the organ level, the tissue level. In addition, there's a lot of research that is suggesting, and more than suggesting, shows clear relationships between stress and cardiovascular disease, AIDS, depression, viral cancers. And when I throw these up, some of you are saying, oh, yeah, and then you'll say, oh, I didn't know that. But the reality is that there's a separation between kind of our common understanding, our general understanding of how stress impacts health, and the data that's out there. When we're trying to look at this in the lab, we're really, really challenged. IRBs don't like us to get people too stressed out. And so we're limited on what we can do. But we do know that animals respond differently. And Selye used all of his propositions for the general adaptation syndrome from animal work. When we start to look at humans, it becomes a lot more difficult. So we have things, though, like the Trier social stress test, which is just public speaking. You bring them in and you have them speak about things that are uncomfortable, and you have an audience there that looks like they're interested in judging them. And we can get physiological activation really pretty easy. But we also have simpler tasks like mental arithmetic and the Stroop color word task. We heard a little bit about the Stroop color word task yesterday, but we use it to perturb or activate the central nervous system. So we're not using it to assess cognitive function here. Roddy did some really cool stuff, and this was in the 1960s. Once the media got a hold of some of the things he was doing in his lab, this was in Great Britain, he had to stop doing his work. And this was the blood work was really cool. He began to say, let's really look at stress in humans. So what he did is he took people that were blood phobic, and then he brought them in, and the first condition was, let's see if we can just have somebody taking blood from another person while you're in the room. The next condition, the same person would come in, and this time They're going to hand them the needle and the tube, and they talk about drawing blood. The next time they come in, they actually draw blood from the person. You can see how it's maybe getting a little more anxiety-provoking. But the fourth one I like the best. After they draw the blood, he asks them to drink their own blood. He did other protocols we can't do that anymore. He did other protocols where he wanted to have people faint. He wanted to learn about this fainting response. And his idea was that actually fainting was quite a beneficial adaptation for humans. It was just the possum, right? It's the possum response. And it might be a survival adaptation that was critical at some point, but he wanted to learn the physiology of it. And he had several protocols that were intended to do that. He threatened fire. He did all kinds of cool things. We learned a ton about the physiology, in particular the cardiovascular physiology from his work, but really very little work was done for many, many years. It was clear that we had these responses, but how do we go further in the lab? And of course, we weren't allowed to do very much with humans any longer. However, I got interested and started to think, well, how about if we combine stressors? Physical is a stress, right? Exercise is a physical stress. Can I add a mental stress to that? Can we then see what might be happening? And then I took a very applied approach to maybe make it a little more intriguing. And we did some things with expectation videos, and I'll show you a study in that area. We did several studies with firefighters where we did a physical stress and a mental stress where they were challenged to make decisions about a fire. And we used firefighters that were what's referred to as incident commanders. So it's the third level of firefighter certification, firefighter 1, firefighter 2. Firefighter, the third level is incident commander. These people, if they're the first on the fire site, they make the decisions. So we wanted to challenge them with a specific drill in how they make their decisions. We then, of course, had to go to Stroop color word mental arithmetic for the normal average population. And then we, again, as I mentioned before, looked at a chronic stress like obesity. In addition, what we began to realize is if we want to learn more, if we really want to get an idea, and if we really think that it might be impacting a cell, we're going to have to really continue to look deeper and deeper and deeper into the system, and that became a real challenge for us. And we also went from looking at athletes, of course, all the way down to looking at overweight and obese, and even aging. Right now we have an aging protocol that's been approved, and we're figuring out how we're going to do some things there. Here's a little background on how it works in our lab. We can do a mental stress, for example, and we're going to activate the brain, right? We're going to know that it's activated by looking at simple cardiovascular reactivity responses. These are real easy, and we can do this in the lab. I can tell you, though, that our perturbations are fairly mild. We're looking at heart rate increases of about 12 beats. If we get a really good study, a really good stressor matched to the population, we might get an 18-beat increase. And other labs are going 8 to 10, so we're pretty good at it, but that's not a super activation. Bet you my heart rate's higher than that right now. Then we went in and we had to look at the catecholamines, right? I mentioned that little diagram, but we wanted to make sure that these were being activated, so we're going to have to get in and look at that. And of course, we're going to have to take a blood sample, but then we wanted to look in particular, I wish it was a little more clear, at something that would have an impact on a physiological response that was linked to health. And so we thought that we would look at inflammation and immune cells. And so we would have to separate out these white cells, and then from those we actually separate out peripheral blood mononuclear cells, because those are the ones that are primarily responsible for producing inflammatory cytokines. All right, so we had to kind of figure all that out over the years, and then we could get to this cell. This is a cell, let's say, barrier here, and we know we have receptors, and then we're going to look at all this stuff to see how it is, not all of this stuff, some of these things, to see how it is that we might be activating an immune cell to produce an inflammatory cytokine that in the end is going to interact with the endothelium level of our arteries to participate in the plaque buildup process. Isn't that amazing? Whew. It only took us 20 years about to get close, and what we have now is better questions than we used to. We don't have many answers, but we have better questions, and so I'm going to take you through that a little bit. When we look at this cell, we were looking at cell receptor expression, transcriptional regulation, and inflammatory responses in particular. So initially, we were looking at this dual stress, combining a physical and a mental so that we could have kind of these different levels of activation. And we knew that epinephrine, norepinephrine, cortisol would be released in a certain manner pattern relative to physiological activation. We also knew that if we just looked at the physical stress, psychological stress literature, it also had some responses. However, in activation that were relatively clear in the literature. However, it was impossible to determine intensity on the psychological side, right? Roddy was the only one to get rid of it, and then he had to get close to it, and then he had to stop because of course, the public attention, and of course, it's much different today. What we first tried was to see if we could perturb athletes. And this was primarily because our lab was a bunch of athletes at the time, and we just jumped in there and decided to pull some in and see if we could do some lab manipulation to perturb athletes. The manipulation we used was an expectation video. They come in under two conditions. They watch a video where there's a runner running at a 70% intensity, not very hard for them. These were sub-elite athletes. And we told them they're going to continue that for 20 minutes. They come in again. They were told it was counterbalanced. It wasn't counterbalanced. They come in again the second time. This time they watch the best runner in the region, 3 or 4 state region. They all know him because they're right at his caliber. And they say, he ran at this is 95% of this person's VO2 max. We're going to put you at 95%, and we're going to see how long you can make it. And if you can make it 20 minutes, that would be unbelievable, right? And this person's dying, sweating on the trail. They're a complete train wreck. And they're looking at that going, wow. What's really intriguing is the athletes, we talked to them, we did interviews afterwards. They thought that this was a brilliant, super cool opportunity to see how they matched up with somebody that was better than them, because we were putting them at their same relative intensity. And we were going to look at time. They thought, whoa, this is really cool. The only reason that bum has been beating me is because he's more physically gifted. Now we're even. I got a shot. It was really, and that was what we were hoping we might get, and it looked pretty cool. So were they stressed? This is just a simple self-report of anxiety that we're getting right on the treadmill, and we suggested, well, they got a little more stressed, we think. Hard to say how much. This is just a self-report. But look what happened to heart rate. It was exactly the same. And then when we went further, we looked at ventilation, it was exactly the same. What in the world? We thought we got them a little anxious. Well, we had known that this might happen. We hoped that it wouldn't, because we would have something really cool to go with. We think it ended up even more cool. We began to look at the literature, and there was a lot written about physiological adaptations. What we learned was, if we looked at how the body adapts, and Dean Spear referred to it at the time as physiologically toughened, we knew that there were physiological adaptations that would occur due to the aerobic training. This was a fit group. Then these could transfer, and this is a really neat proposal on how this works, and it makes a lot of sense. Softman thought that it could transfer into some psychological benefits, so that what would happen in the end is you would have a more emotionally stable response. And if you did, physiologically, you would have a more stable response as well. This helped explain our data pretty well. So of course we had to go back and look at some unfit individuals. And so of course we went back in. We did a nice next study was to look at fit and unfit individuals. They were actually pretty close in fitness level. And this time we actually started in the lab by trying to show them a video and perturb them. But these were individuals that aren't competitive, right? They're your average, normal kind of run-of-the-mill human. And we didn't understand that. Remember, we're all just a bunch of running endurance geeks hanging out in the lab. And we started showing videos. They're like, what are you doing? So we had to learn about things like the Stroop color word task and mental arithmetic to try to perturb them. Now, interestingly enough, we went back and looked at the literature, and psychologists had been doing mental arithmetic, Stroop color word, for some time to look at stress. But Roddy, in his description of his protocols, talked about harassment. They would be insulting the individual pretty directly on their abilities to do math. And so we came up with something called critical evaluative feedback, so a series of comments that we used to ensure that we might get them a little more focused, a little more, let's say, of course, anxious about the circumstance. In the end, low-fit demonstrated a greater increase in heart rate response to the combined mental and physical stress, and this was further support for Dean Spear's model. We've done another series of studies in this area, and it's pretty clear, hard to tease out, but we can blunt this, let's say, heart rate response, and it's coming off of some training. Now, there might be a ton of other explanations, and we write about those, and they're likely all interacting, but it still was kind of nice to see this data. Heather, a doctoral student in the lab, she'd been in our lab for about 3 or 4 years, and she decided she might want to graduate. And we had thought we needed a critical population to look at. We needed to have an impact on a population of interest. Well, remember, when we looked at the athletes, well, they had the blunted, they had a physiologically toughened response. So we couldn't help athletes that much with coping with stress. But there was another group, and it was firefighters, and she spent a lot of time in this work. Firefighters have one of the highest rates of on-the-job heart attack deaths among all occupations, and heart disease is the primary cause of line-of-duty deaths in firefighters. In addition, multiple studies have shown that firefighters have increased mortality ratios of ischemic heart disease compared to other cohort populations. Now, maybe this is due to this dual stress, right? Doing physical activity in a high mental stress environment. So were there things that we could do in the lab to kind of manipulate some of these variables? And there was. This is a firefighter strategies and tactics drill. And the individual, the firefighter, remember, an incident commander, is presented with a fire and a series of questions that they have to address relative to the fire. While we're collecting our data, we've got a catheter in there, we're drawing blood, doing all kinds of things. The task itself we got from the National Firefighting Institute in Bethesda, Maryland. We went up and visited. They had never had a computerized one. We made it computerized so we could use it in our lab, but they had done it through a paper and pencil. Of course, we told the firefighters that their fire chief would be getting the results. That we couldn't do that, really, but we were able to at least deceive them while they were in the lab. So we hoped that we would get some kind of activation, and indeed, when we looked at our data, we were quite effective in elevating heart rate in the dual challenge condition versus the exercise alone condition, respiratory rate, ventilation, VE, VO2. It suggested to us that when you're battling a fire, you've got dual stress going on, and it's having a physiological effect. Of course, we had to go back in and say, okay, let's take a look at some stress hormone responses as well, right? That's the next level of understanding that we wanted to look at. And this is consistent with all our stress work now. When we look at, we're going to have to keep them in there for a couple of hours. We got to get a lot of good baseline work. And then when we look at the firefighter strategies and tactics condition, compared to the exercise alone condition, we always get these elevations, I should say. It's not always. Epinephrine can kind of be a little funny for us, but norepinephrine we get in every study. Of course, we wanted to look at the HPA axis, or cortisol, as well, and sure enough, we get a nice cortisol response in exactly the way that we might expect it to occur. Of course, the individual is working at an exercise intensity that we would predict would not cause cortisol to be elevated. We do get a slight cause, but increase, but at least not significantly. This was really cool. We now had something that might be pertinent to a specific population, and we ended up doing a number of studies in this area. We started to look at the immune system activation overall. Let's go back in and see if their immune system is activated, and sure enough, and this was Phil, another doctoral student that was now overlapping into our group, just got crazy about the immune system, and I learned a lot from Phil. I don't think I ever took a course in immunology, so with Phil, and Phil was originally from Taiwan, he would come in with a PowerPoint and a book, and we would together, while he was taking courses in immunology, and we would together go over it, and it actually was a pleasure for me to learn that way. It was really fun. But Phil is now really knowledgeable. We saw activation of the immune system. This was important. This was a more general immune activation. Now we wanted to go after those peripheral blood mononuclear cells to look at the inflammatory response, right? That was the next level. We wanted to get there because we knew that's where maybe the best story or the most important story in how this is happening physiologically. And so we were really looking at this inflammatory response and understanding that it might link, or it does link, to endothelial dysfunction and, of course, cardiovascular disease. We spent a long time trying to figure out what the best way would be to do this in terms of measuring exactly what we wanted to measure. We knew that these inflammatory cytokines, there was a lot of work on inflammation and targeted interleukins that we would want to look at. And so we began to figure out, what are we going to do and how are we going to measure this? When we looked at the inflammatory cytokine work on dual stress, or I should say on simply psychological stress, the results were all over the place. Equivocal, bouncy, different labs finding different things. We weren't convinced that we were going to be able to perturb the system and actually look at these cytokines, and we were quite worried about it. What we ended up doing is saying, maybe we should be a little slower, thank you, than we are, and slow things down and take a little closer look. Now, this is just the mechanism that we're looking at in how it interacts with this endothelial level. But here's our pro-inflammatory monocyte. You can see that there's a ton going on here that I'm not going to talk about today, and we don't look at all this, and it's even much more complex than this. But we do know that if I elevate this, that it is and does participate and is related to all of these other things happening in the plaque buildup process. Then we stumbled into a study, I think this is 2009 if I'm not mistaken. Prather did really cool work. And what he was interested in was some gender differences, and he looked at premenopausal and postmenopausal women, compared them to men, baseline task recovery. This is just psychological stress. This was the cleanest data we'd ever seen. But what he did with the cells, the immune cells in particular, is when he took them out, he actually stimulated them again. Remember, our heart rate and his heart rate increases were about 10 to 12. We were doing a little bit better than that, beats per minute. What he was able to do is he stimulated the cells further in a tube. And when he did that, he was able to clear up some of the data, the inconsistencies in the results that had been presented in the literature. So we knew that's what we needed to do. We needed to stimulate our cells further in a tube after we withdrew them, and then look at the mechanisms of interest. Now, that doesn't mean that that's exactly what's happening in the body, but in doing that, we could at least begin to look at what might be happening at higher levels of activation. So Phil came back and said, not only are we going to do that, but we're going to start to look at obesity as a chronic stressor. So we're now going to just sit them in front of a computer and activate them, but we can look at lean and obese responses, for example. And that's exactly what we started to do. And we're going to stimulate ourselves so that we can look inside the cell and see what might be happening. So this is a standard look of our lab and kind of the things that we do in putting a subject through our protocol, and then, of course, all the analyses that occur after that. This is a really cool series of results in that when we stimulate them with this lipoprotein saccharide here, we see an mRNA. So now we're looking in the cell, and we're looking at, did we activate the synthesis of the pro-inflammatory cytokine? That's what we're looking at here. That's different than looking at the protein that's already been synthesized. We find it either in the blood or in the sample, because that's the protein that's already produced, right? This is mRNA initially. Both the lean and obese responded similarly. However, I'm pointing out here in this slide, this is the 24-hour activation, and sure enough, we see the obese activate this pro-inflammatory cytokine much greater. We guessed at 24 hours. We thought we still need to be careful here. We could have gone 8 hours, 12 hours. We ended up going back and looking at that data, but the 24-hour data gave us the clearest result. It's just telling us that the obese individuals respond, these cells are responding differently. This is pretty intriguing and in a more pro-inflammatory process. But we also have to appreciate the anti-inflammatory responses that are also occurring. It's an inflammatory milieu. So what we ended up saying is, okay, there are a number of factors in this obesity side that are different than, let's say, the normal weight side, and we should expect different responses all the way down to gene expression and, of course, down to the risk of cardiovascular disease. Heather Caselin in our lab came out next and said, I'm interested in leptin. I'm interested in this unique hormone related to obesity. And she showed that, sure enough, this is an anti-inflammatory response. And for the obese individuals, leptin was correlated to this anti-inflammatory response in the non-obese but not the obese, giving more evidence that the physiology and how these cells are responding is different. Phil came back and showed us that, sure enough, if we continue to look at mRNA versus the cytokine protein response, we're going to see mRNA responses before we see protein responses. This is going to be challenging. We're narrowing it down. We're looking at obesity. We do find correlations with obesity. Again, it's now how do we go anti-inflammatory, pro-inflammatory, activation, deactivation. All of these stories are getting much more interesting. What this last study looks at is this GRK2. This turns off the process. And sure enough, that's activated as well. And it seems to be activated greater in the lean than obese. The story of what's happening in the cell becomes far more complex. And all we've done is come up with better questions. The future, it's all about understanding this milieu. It's all about trying to figure out what the mechanisms of activation are, right? And also this timeline for synthesis versus deactivation. For us in our lab, that's our future. If you look overall, this is my last slide. Well, except for the thank you. It's how much, how often, how long, what type, and for whom in terms of physical activity. It's physical activity and medicine. So what we're trying to do in our next series of proposals is to say, if I add physical activity to a medication, and the medication is deactivating or activating pro or anti-inflammatories, long story, what effects should the exercise have with the medication? Maybe I can take less of the medication if I exercise, in particular if the activation process is different. Males and females seem to be different. Genetic influence, stress in the microbiome. I have one student who's going crazy right now, and I've got a lot of work to do there before I get there. These folks have been critical. They're colleagues at VCU that has helped us to continue investigating this process. Thank you. I did it.